Hello, this is Ed Chapman, and this is part 4 and the last part of the series covering metabolism, how enzymes work to store, release, and use energy inside of cells. If you remember, we first talked about enzymes as being a lot like highways along which things travel. Well, the stuff that's traveling along metabolic pathways are chains of reactants and products. And let me try to show you what I mean. Imagine you have a raw ingredient that you're starting with, and this raw ingredient is going to be the substrate for an enzyme, and the enzyme is going to change it and produce a product. Okay, but that isn't where it stops. That product then becomes the reactant for a second enzyme, and you get a secondary product produced. And then let's say that secondary product becomes the reactant for a third enzyme, and that's what produces your final product. So in order to get to the final product, we had to go through 3 different enzymes. Now, why would nature design systems like this? Why couldn't you just have an enzyme that takes the initial substrate to the final product? Well, it may mean there's a lot of different activation energies here. It may mean that we need to regulate this at different locations. It may mean that this enzyme pathway is connecting with other enzyme pathways in kind of a branching pattern or a converging pattern. There are 1,000 different ways this could be interpreted, but this is the way cells normally operate. It's the interaction of lots of different metabolic pathways controlled by and actually made of different types of enzymes. All right. So let's look at how enzymes can be regulated, which means activated or deactivated. All right. So enzymes, when they're regulated, they can either be switched on or activated or turned off. In other words, deactivated by other molecules. And the first and probably the easiest to understand is competitive inhibition. This is when there is a molecule in play called an inhibitor, which can also fit into the active site of the enzyme. So the substrate can fit in here, of course, but this inhibitor can also fit in here. And this means that this molecule and this molecule are competing for the active site. That's where the word competitive comes from. Now, if the substrate gets in there, then the enzyme functions normally and you get your product. But if the substrate gets in there, if the inhibitor sticks in the active site, then you don't get the final product. Nothing happens, okay? Absolutely nothing, because the enzyme is effectively shut down. And this effect generally lasts, until the body is able to remove or break down the inhibitor. Now, the cool thing about competitive inhibition is it's the model that scientists believe explains the function of a lot of painkiller drugs. Painkiller drugs generally work by blocking nerve signals, and the way they block nerve signals is that they're able to fit into active sites on nerves and stop the normal function of these embedded enzymes stuck in the membranes of nerve cells. Okay, so these competitive inhibitors are able to lock into these sites of the receptor proteins in nerve cells and stop them from working. So even though pain may still be occurring, the brain can't, quote unquote, see the pain or feel the pain. So as far as you're concerned, the pain is gone, at least temporarily. And as I said earlier, this lasts until the drug breaks down or the pain receptor proteins are replaced by new ones. Okay, so I just highlighted here the receptor proteins, which are kind of functioning like membrane-bound enzymes that can be competitively inhibited by the presence of a painkiller drug like Novocaine or cocaine or any of the AIM drugs out there. They're all nerve deadeners, and they work by blocking the function of nerves. Okay? All right. Another type of enzyme regulation is allosteric regulation. And there can be allosteric inhibition or allosteric activation. This diagram shows allosteric activation. And the way it works is there is an active site that a substrate can fit in, but the active site doesn't have the right shape unless another location on the enzyme is filled by an activator molecule. All right, so the activator molecule has to bind with the allosteric site to turn on the enzyme. Okay, so if this little guy is able to get in here, then notice the shape of the active site changes in a way that allows the substrate to fit in. And once that happens, then you can get your enzyme substrate complex, which can then react in whatever way it's going to react to produce a product. Okay, so we have a functional pathway as long as this is bound. Lots of vitamins. You hear about vitamins and minerals and things like that. Lots of vitamins function as allosteric activators of enzymes. So your cells have to have a certain amount of these vitamins present in order to function normally because they are activating the enzymes. All right, clicking through. All right. Another type of allosteric regulation is non-competitive inhibition. It's pretty much the same idea as what we just looked at, except in this situation, there is an inhibitor that can fit into a site. Okay, so here we have a normal situation where the active site is not filled by the inhibitor. Excuse me, the allosteric site is not being filled by the inhibitor. Excuse me, I completely messed that up. Okay, let me get my drawing going here. Okay, this isn't the best diagram, I'll completely admit that. But here you have an enzyme that's able to receive a substrate and change the substrate into products. These little things here represent the products. Okay, now down here we have the allosteric site. Now up in this example up here, the allosteric site has not been filled by the inhibitor. Okay, here they're calling the allosteric site the inhibitor site. Means the same thing. In this example down here, B, that's trying to show non-competitive inhibition, here the inhibitor has actually docked into this allosteric site. And when that happens, it causes the enzyme to change shape, change its conformation, and now the active site isn't going to work anymore. And in this particular example, they have the substrate going in, but it's not going in the right way. So it comes out unchanged. So this isn't the product, this is just the substrate unchanged. So in this situation, we have an allosteric site that if it gets filled by an inhibitor, it deactivates the enzyme, so the enzyme doesn't do its job anymore. All right, hope that wasn't too bad. Okay. Finally, we have another form of regulation in enzyme pathways called feedback inhibition. And I think this is the coolest one because here you have a situation where an enzyme, and pay attention to the enzyme shape and number here. Enzyme 1 can receive a substrate and change that substrate into a product. Okay? That product can then fit into a second enzyme. This is enzyme number 2, and get changed into a final product. Okay? Now this final product can fit into the same enzyme we started with. And if this guy gets into the active site, if the final product here gets in the active site, this enzyme pathway is shut down. Do you see it? The final product of the entire pathway is an inhibitor, a competitive inhibitor for the first enzyme in the pathway. So this is a great way for an enzyme pathway to self-regulate because once there's enough of or a high enough concentration of the final product floating around in the cytoplasm or wherever this might be happening, then the system shuts down so that you don't make too much of it. Okay? So we call this feedback inhibition because the presence of the final product is feeding back on the beginning of the enzyme pathway, and it's acting as an inhibitor because it's deactivating enzyme number 1, which starts the whole process. Okay, hope that makes sense. All right, stop there. We are finished with metabolism, and thanks for listening. Oops.